Hello, this video is on photosynthesis and includes the required practical 5 from GCSE Biology. As usual, you will need a pen and a piece of paper if you want to participate properly in the video, because every now and again I'm going to ask you to pause it while you complete an exam question or fill in some blanks or whatever it is that you need to do. So if we're looking at this, we've got a few bits to do with photosynthesis. There's some things that you're expected to know regarding photosynthesis. The equations are obviously a big part of that because you will be asked to link them into other potential questions. The uses of glucose does not come up at in this year's GCSE. It's not on the pre-release information, so it's not in my PowerPoint either. All right, we're not going to look at that one. The required practical definitely is on the paper because they have put that on the pre-release information. So we'll be looking at the variables of the required practical and then any limiting factors of photosynthesis as well, any issues that are going to reduce or increase the rate of photosynthesis as a reaction. So the photosynthesis equation usually looks a little bit like this. We've got water plus carbon dioxide producing glucose and oxygen. The reactants being on the left-hand side, that would be water and carbon dioxide, and glucose and oxygen, the products, being on the right. Occasionally you might see this with the word sunlight above the arrow in the middle. That's absolutely fine, but don't write that in your answers if you get asked to fill in the blanks in an equation. That won't be what's missing. So yeah, I wouldn't include that one as part of your photosynthesis equation. So we've got our reactants, we've got our products. This is a symbol equation. For those on higher tier, it's more likely to come up for you guys than it will for foundation. Water is H2O, carbon dioxide is CO2, glucose is C6H12O6, and oxygen is O2. And if you wanted to balance this, you'd need 6 H2Os, 6 CO2s, and 6 O2s. We don't do anything to the glucose at all. Okay, so we have a little bit of independent practice here. We've got some missing words to do with photosynthesis. If you want to pause the video at this point to fill in the blanks, you can then play again, and I'm going to go through some of the answers with you just to check and make sure that you are happy. Okay, so answers for this one here, you can have a read of them off here. Carbon dioxide and glucose in the first one, it's 6s all around for water, carbon dioxide and oxygen on the second one. Nothing happens to glucose, we don't put anything on glucose at all, C6H12O6. The reactants of photosynthesis are carbon dioxide and water. The products of photosynthesis would be glucose and oxygen. Chlorophyll is our green pigment, light energy is needed, and the chemical formula for glucose is C6H12O6. Okay, so the photosynthesis required practical, we have been told, will be on your exam. We're going to talk through some of the variables associated with it and how you would carry out the practical. You're meant to know it reasonably well. We have some questions on it to have a try of in a little while. So the hypothesis in this case is going to be about light intensity. Okay, as the light intensity increases, the rate of photosynthesis increases. The closer the lamp is to the pondweed, yes, this is your lamp and your pondweed practical, all right? The closer the lamp is to the pondweed, the more bubbles of oxygen or the higher volume of oxygen should be produced. So our independent variable is going to be light intensity. Okay, that's the thing that we are choosing to change. The closer we put the lamp, the higher the light intensity will be. The dependent variable is going to be the rate of photosynthesis. So we are trying to measure the rate of photosynthesis using either counting the bubbles or measuring the volume of gas produced with an upside down measuring cylinder. And finally, we have control variables. Anything that's going to affect the rate of photosynthesis that is not light intensity has to be a control variable. So the species of plant, if we have a different species of plant that photosynthesizes faster, we can't swap to that one halfway through. Okay? If we have a different color of light for the lamp, and the different colors are going to make photosynthesis happen faster or slower, then we can't suddenly swap to green when we've been using blue. The temperature matters, and obviously your lamp is going to get hotter the longer you leave it on for. We do not want the temperature of the pondweed and the boiling tube that it's in to change. We'd like it to stay the same. Just adding a thermometer to this will not control the temperature. So if you're asked how you control the temperature, do not say adding a thermometer, because although that will tell you that the temperature is changing, it will not stop the temperature from changing. The concentration of carbon dioxide is also important. We usually add something called sodium hydrogen carbonate, a chemical that will provide carbon dioxide for the plant, rather than water. Because in that case, we know that the carbon dioxide level in that tube is as high as can possibly be, and therefore it is not a limiting factor of photosynthesis. It's not slowing anything down. So we're going to have a quick look at a method, okay, of how to do this, although you guys have done this in lessons already. We would set up a boiling tube with 0.2% sodium hydrogen carbonate to provide carbon dioxide for our piece of pondweed. We're going to cut a piece of pondweed to a specific length. It doesn't have to be 8 centimeters. 8 centimeters is a number that we've put in here. However, if they're all at 8 centimeters, then they're being controlled. The size of the piece of pondweed, the length, and therefore the surface area of the leaves is being controlled. And we insert it into the boiling tube, ideally with the cut end up over. We're going to place the boiling tube on the top of a beaker of water, and place a boiling tube on the top of a beaker of water and flip the boiling tube upside down, so that your pondweed is underneath the water. Ideally, this would mean that the boiling tube is going to be absolutely full of water at this point. We can see that the boiling tube's there. I think when we've done this in class, we've used a measuring cylinder instead, because it's easier to measure the volume of gas, and that would be an improvement to this practical over just using a boiling tube. What we've then got in the bottom of there, we've got our piece of pondweed underneath a funnel, so that any bubbles coming out of the pondweed is going to go up through the funnel and into the boiling tube. And basically, we're going to count the number of oxygen bubbles in this one here. Your final equipment setup should look like the image on the right-hand side here. You're going to set up your lamp at a particular distance away, and then you are going to count the number of bubbles per minute. Remember that you need to leave a period of time here for the pondweed to start photosynthesizing. If you start counting straight away, your first few results for whatever distances you use are going to be 0. Okay, we need to leave it until the pondweed started photosynthesizing. Once it has, then we're going to start counting, and we're going to count bubbles per minute. Okay, remember, you don't actually have to count bubbles for an entire minute. You could count bubbles for 15 seconds and times that by 4, or count the bubbles for 10 seconds and times it by 10, but you are going to get a less accurate number of bubbles at that point, because you haven't counted for quite as long. We're going to record the results, and we're going to move the lamp to the next distance. The amount of people that forget to talk about moving the lamp when they're talking about carrying out this practical is crazy. Okay, so make sure you have talked about moving the lamp to a different distance and testing again for the same period of time. You're going to repeat at every distance that you do. If you do 5 distances, that would be 3 repeats at each of those 5 distances, and calculate an average number of bubbles for every distance. So there are 3 limiting factors of photosynthesis that you are meant to know about. Technically, there's 4. The 4th one we'll talk about in a second as to why it probably shouldn't really change. Temperature, light intensity, and carbon dioxide concentration all affect the rate of photosynthesis. So does the amount of chlorophyll, But if you're using the same plant every time, then the amount of chlorophyll will not change. If you're using different plants, there would be absolutely no way of controlling the amount of chlorophyll. You could not stop that from changing. In different plants, there will be different amounts of chlorophyll. Temperature, light intensity, and carbon dioxide concentration can all be changed or controlled. Okay, you will notice on the graphs that for light intensity and carbon dioxide concentration, as you increase carbon dioxide concentration, the rate of photosynthesis increases. That's exactly how you should write it in the exam. That happens up until a particular point where it starts to level off. So again, in your exam, you would write that increasing the carbon dioxide concentration beyond that point will not increase the rate of photosynthesis. It will stay constant. Okay, but we'll talk about what's on the axes. As the light intensity increases, the rate of photosynthesis also increases up until a certain point. Beyond that point, increasing the light intensity does not increase the rate of photosynthesis. Temperature is slightly different because for temperature, we're talking about enzymes. Photosynthesis is an enzyme-controlled reaction. So as the temperature increases, so does the rate of photosynthesis, but only up to that point in the middle. It'll be about 40 degrees-ish, maybe 45. Beyond that point there, increasing the temperature will decrease the rate of photosynthesis because the enzymes are becoming denatured. If you don't know what that means, you might need to go back and have another look at enzymes. So, couple of practice questions for you. I'm not going to talk you through all of these, but I am going to show you the mark schemes. These are going to be the points where you're going to probably start pausing the videos and writing down your answers. So for the following hypothesis, write down the independent, dependent, and control variables. We've got our hypothesis here. As the temperature increases, the rate of photosynthesis will also increase to an optimal point. You need to write down the independent, dependent, and control variables. That would be the thing that you have chosen to change, the thing that you are writing down as your results that you're measuring, and things that need to be kept the same. Pause the video and have a go at that now, please. Okay, so answers for that one. The independent variable would have to be temperature because that's what we've said we would change. The dependent variable is therefore going to be the rate of photosynthesis, the same as it was in the experiment that we looked at before, and we're going to count the number of bubbles produced per minute. Finally, control variables could be light intensity. We don't want to change the light intensity if we are changing the temperature, and we don't want to change concentration of carbon dioxide if we are changing the temperature. We can use sodium hydrogen carbonate to keep the concentration of carbon dioxide the same. Okay, one more. So this is the method used. This is another easier one, this, so they've given you a little bit more structure. There's a short method, only 4 steps, and then you have to suggest 2 ways the method could be improved to make the results more valid. Again, pause the video, have a go, and I will do some answers in a sec. Okay, so answers for this one. You could repeat and calculate a mean because a mean is going to be much more representative of the real number. It's going to give you a more accurate result than if you just have a one result at this point. Or you could repeat it to eliminate anomalies. So if you've got 25 bubbles, 30 bubbles, and 26 bubbles, and then one of them was at 4 bubbles, the 4 bubbles is probably wrong, and we can probably get rid of it so it doesn't spoil our average, our mean. You can control the water temperature. You can control the carbon dioxide concentration. Controlling water temperature could be done with a heat screen, plastic screen, or with a large beaker of water, or it could be done by using an LED bulb, which means that it won't produce any heat. You could control the carbon dioxide concentration by using sodium hydrogen carbonate. We can control the distance of the bulb from the pondweed, set it up at the right distance. Okay, there's not a lot to do with that one. Control the mass, length, species or age of the pondweed. Use the same piece of pondweed, covers all of that. And give pondweed the time to equilibrate, okay, to get started properly. So you can do the experiment with the bulb off or in the dark first to show what would happen if there was no light, possibly. Okay. So last one, and this one's slightly more complicated. This one here is just giving us 4 pieces of equipment, and it suggests that you should set up this practical. How would you set up the apparatus? Now, I always think this one's a bit mean because this is actually only worth 1 mark out of the 6, okay, for setting it up and just talking about how you would put it together, but it is still worth doing that. The other marks all come from the measurements you would make, so that's your independent, dependent, and control variables. And again, control variables, how could you make this a fair test? So for each one, you need to talk about how you would control them. I'm going to let you pause the video and have a go at writing a method, including all of your variables, and then I'm going to do some answers. So, answers for this one here. The apparatus setup is worth a mark. Weed in the water in the beaker and light shining on the beaker. That's the idea in there. It's just setting it up correctly. Method of varying the light intensity is our second one. So how do you actually change the light intensity? And according to the banding on this question, which I haven't shown you on here, if you don't talk about varying the light intensity, then you cannot get any more than 2 marks out of 6. Okay, so you have to talk about how you would vary the light intensity. It's very easy. Change the distance of the lamp from the plant. Any methods for controlling other variables get you a mark. So using the same pondweed or the same length of pondweed, water bath or heat screen, CO2 by using sodium hydrogen carbonate, leaving it a sufficient amount of time gets you a mark. So making sure you don't just set it up and set it straight off. You have to leave it a little while for it to equilibrate. Okay, a method of measuring photosynthesis. So you could either have talked about counting bubbles or measuring the gas released. Measuring the rate of photosynthesis by counting bubbles for a set period of time. So this is rate. And without mentioning rate, you can't be in the 5 or 6 mark band. It's about how many bubbles or how much gas was made in your 1 minute or in your 5 minutes. It's a rate, not just a number. And any repeats gets you a mark as well. So this one here was just looking at the results table and the possible results table. So this is an example results table for this. You can see all the different temperatures down the left hand side and all of the different results that they have got. They have calculated means for everything but one. You could pause the video here and calculate a mean if you wish. So again, we've got a couple of questions about the practical. Suggest one possible cause of the anomalous result that was in the table, and how could students deal with that anomalous result? What have they actually done with it? And again, I'm just going to go through answers on this one. You can't see the stuff without sending your video back over, so we'll just have a look. Their scale might have been misread. There could have been air or oxygen in the syringe. The lamp or the light might have been moved. The temperature might have changed. They could have had a different mass or length of pondweed if they've changed their piece of pondweed halfway through, or they might not have left it to acclimatise at that point, so they've got a result that doesn't work. How did they actually deal with that? Well, they just didn't put it in their calculation of their mean, and that's what we always advise you to do. If you have an anomalous result, just don't put it in there. Okay, so that's the end of our photosynthesis video, and thank you very much for watching. Good luck with your revision. Take care.